whatever I was talking, my whole thing was surrounding the glucose, right? I have taken your glucose estimation in the urine, then the discussion of the diabetes mellitus and all the related defects of the carbohydrate metabolism and all, everything was covered. So among all of that, you people got to know that we have to first estimate the blood glucose level in the urine, or the reducing sugar in the urine, as well as the glucose level in the blood as well, right? So today's topic will be how to estimate the blood glucose level. Okay, when we are measuring the blood glucose, we are not exactly measuring the blood particles, we are measuring the plasma or the serum. Okay, so we are taking the blood sample, we are centrifuging it, the RBC or the particles, whichever is present in the blood, that is precipitating at the bottom, and only the clear plasma and serum is the supernatant. From the serum we are taking the sample, then we are measuring the blood glucose level. Okay, now there are certain things that how we are doing the test, that is one thing which is important. The theory class today will be very short and on the point, whichever is necessary, how to perform the test, and that's all. But one thing that you have to keep in your mind is you have to good with your pipetting. Okay, so estimation of the blood glucose by GOD-POD method. The full form of GOD-POD is glucose oxidase peroxidase method. Okay, in short we say GOD-POD, but if the examiner is asking, don't say GOD-POD, say glucose oxidase peroxidase method. Okay, this instrument you know, everyone is acquainted with this instrument. Name of the instrument? Colorimeter. What is the law on which it acts? The law? Just now I said Beer's and Lambert's law. Okay, what is the Beer's law? Okay, A directly proportional to C. A is the absorbance, C is the concentration. Okay, and the Lambert's law? Directly proportional to the length that the rays are covering, right? Okay, so that is A directly proportional to L, right? Together we say it A directly proportional to C and L, right? Now over here we are using the cubettes, and the cubettes L is fixed. All the L's are same, so that is why we come to the conclusion A is directly proportional to the concentration. Okay, so the absorbance is directly proportional to the concentration of the particles that is present in the solution, right? Okay, fine. Now by that same principle only we are doing over here. What we will do, we will take some samples, we will put it in the calorimeter, and we will see the absorbance. If it is containing glucose, then the absorbance will show how much amount of glucose. The more the glucose concentration, the more will be the absorption value. Okay. Okay, let's get into the topic. First of all, introduction, sample collection. Plasma is the preferred sample for estimation of glucose. Okay, we all know whenever you are estimating the blood glucose, you cannot estimate from the blood particles. You have to first centrifuge it, take the serum or the plasma to estimate the blood glucose from it, and that is the circulating blood glucose. Okay, now which kind of blood container? Now when you will go to pathology, that time you will see there are different color coded vacuitainers. Vacuitainers are what? The blood containers where we collect the blood, the sample where it is collected. Okay, if you see in some lab, if you have given blood, this is a container, okay, and this is the cap, and this cap is different colors. Okay, these caps are having different colors. For blood glucose to estimate, we have to take the blood in a vial which is having the gray color cap. Okay, now that is also have a reason. Why gray color cap only? Why not a orange or a blue color cap? Why not a green color cap? Okay, because these containers already contain some chemical particles, fixed chemical particles as per the coded colors. This gray color contains, one is the sodium fluoride, another one is the potassium oxalate. Okay, these two things are there already in the vial. Now what is the functions of the sodium fluoride and the potassium oxalate? You all know, if we take blood from a patient and keep it in an open space, after some time the blood will coagulate, right? That is blood's normal property. Now if we add potassium oxalate into it, the blood will not coagulate. It will remain in the liquid form only. That is known as an anticoagulant. It is not letting the blood to coagulate. That is why it is known as the anticoagulant. So the potassium oxalate's function is as an anticoagulant. Okay, so that as soon as you are taking the blood should not coagulate after some time. Okay. Second thing is sodium fluoride. If you recall from your theory, the glucose metabolism pathway, in the glycolysis, there is one step when the enolase is the enzyme which is causing the glycolysis, right? Now sodium fluoride causes what? It inhibits this enolase enzyme. Okay, so because of that, when you are taking the sample in a container, automatically some bit of glucose will undergo glycolysis. Okay, so just to avoid that, you are adding sodium fluoride so that glycolysis should not happen. What will happen if the glycolysis is happening? The glucose value will be less, because the glucose is already getting broken down, right? Glycolysis is happening. So to avoid that, you are giving sodium fluoride into the vial already. So that is how these vials are already, these chemicals are already there inbuilt. Okay, and the function of the chemicals are, one is the sodium fluoride, which is helping inhibiting the glycolysis at the enolase enzyme step. Okay, and the function of the potassium oxalate is it acts as a anticoagulant. Okay, any doubt? Okay, so these two are the things that is present in the gray vacuum thinner. So questions can be asked like, if you are going for blood glucose estimation, which color coded container you will take? Gray color code. What are the constituents? One is sodium fluoride, another one is the potassium oxalate. What is the function of sodium fluoride? It is inhibiting the glycolysis at the enolase enzyme step. And what is the function of the potassium oxalate? It acts as an anticoagulant. Done. Now, blood should be collected from the veins because in fed condition, arterial blood sugar may be higher by 35 mg per deciliter. Now, what do you mean by fed condition? What is fed condition? After having food. Okay, what they are saying that blood sample should be collected from the veins, not from the arteries. Okay, in fed condition, the arterial blood sugar may be higher by 35 mg per deciliter. Why? Anybody has any question or any answer to this? Yes, exactly. So what he is trying to tell is, let's say you took food, now from the intestine your glucose got absorbed. Okay, that is coming into the arterial circulation. Now what it will happen, all this circulation is coming and going into the heart. Heart is pumping the blood and the bloods are going to the periphery. Let's say this is my fingertip. The blood is coming to the fingertip, okay, with reach in glucose. Now in the periphery, the tissues are doing what? Taking up the glucose from the blood, whatever they require. Like that, every organ and every peripheral part of our body is taking up the glucose from the blood. And after taking whatever they require, rest of the remaining glucose is going back via veins. Arteries going towards the periphery and veins are coming back from the periphery. So veins are coming back. So obviously veins will have less blood glucose as compared to the arteries, because arteries are taking the blood glucose before being utilized. Okay, our veins are carrying after the peripheral tissues are utilizing the glucose. That is why we don't take blood from the artery to estimate the blood glucose. If we take, we will get a falsely raised blood glucose level. Got it? That's all. That is what is written over there, that the blood should be collected from the veins because in fed condition the arterial blood sugar may be higher by 35 mg per deciliter. Okay, so which is the most common vein from where we take the blood? Median cubital vein. That is the most commonest. Okay. What? Third point. See, you are taking food. Your food is going to the intestine. It is getting metabolized, and there are some transporters which is absorbing the glucose from the intestine, right? Like SGLT1, SGLT2, GLUT4, GLUT5. These are absorbing the glucose. Then after that, these are going into the blood circulation. This blood circulation is coming back into the heart. From there, these are getting pumped. Once they are getting pumped, this glucose rich blood will go to the peripheral tissues. Now peripheral tissues, let's say I gave an example of my fingertips. My hand also, over here also. Everywhere, all the tissues will take up some bit of the glucose from the circulation. And rest, there will be some bit of glucose remaining in the circulation. Let's say in the artery, after your digestion of the food, you got around 150 mg of blood glucose. Now this blood will get transported into the multiple parts of the body. Let's say liver, kidney, heart, brain, everywhere all the bloods went. These took 10, this took 10, this took 10 mg, this took 10 mg. Just an example. Everybody took 10 mg of glucose from the circulation. How much is remaining? 110. This will go back to the heart again via veins. And this will keep on circulating. Right? So this value is after the utilization of the organ, and if you take this is before utilization of the organ. Okay, so this is a false reading. Why? Because this much amount will get used up by the organs. You are still adding it, so that is a falsely raised blood sugar level. That's all. Okay, now there are various methods of estimating glucose. Okay, One is the enzymatic method, that is the most important method, and that is what we are doing over here as well. That is glucose oxidase method, also known as the glucose oxidase peroxidase method. Okay, and then hexokinase method. These two are the enzymatic methods. Okay, like that reduction methods are also there, like ferricyanide Hoffman's method, copper sulfate method, and aromatic amines method. Okay, chemical tests are there like modified King-Asatur method and orthotoluidine method. And other tests like Clinistex, Diastex, and the capillary blood glucose test. Okay, these are the types. Okay, and these are the examples of those types. Okay, now in exam they can ask, other than glucose oxidase peroxidase test, what other test can be done to estimate the blood glucose level? So you can mention these names. Okay, if somebody is specifically asking what are the enzymatic methods, then only these two will be your answer. Okay, so if they are specifically asking, say glucose oxidase peroxidase method and hexokinase method, these two are only enzymatic. If they are going for chemical test, modified King-Asatur test and orthotoluidine method. Like that you have to answer. If somebody is asking what are the tests, you can say any name. Okay, if they are being specific, your answer will also be specific. Okay, now over here we will be performing only the glucose oxidase method. Okay, rest of the method we will not be performing. Why not? That I will tell in the later on. No, you don't have to do all of this. You have to just remember the names. That's all. If somebody is asking what are the methods, the first answer should be glucose oxidase method. Then you can say hexokinase, modified King-Asatur, orthotoluidine. After that you can remember, you can say. Okay, these four are mostly important ones. Okay, but we will be doing only the glucose oxidase method. Let's see. In glucose oxidase method, what are the reagents we are using? First of all, the enzyme reagent. Enzyme reagent comprises of four things. One is glucose oxidase, peroxidase, 4-amino antipyrin, and phenol in the phosphate buffer. There are enzyme reagent. Enzyme reagent comprises of four things. One is the glucose oxidase, peroxidase, 4-amino antipyrin, and phenol in phosphate buffer. Okay. Then there is glucose standard. Glucose standard means you know the concentration of the standard, how much glucose it contains. It contains 100 milligram per deciliter. That's a fixed amount. Okay, that is given to you. This enzyme reagent and glucose standard both comes as a kit. Okay, you open the kit, you see it. Okay, and that is how it is written over there. Now what are the compositions? That is written over there, and the functions of the compositions I'll come. And test sample is also there. That is the glucose concentration you don't know, unknown sample. Okay, now what is the functions of those reagents? First of all, there is glucose. Glucose is being oxidized by glucose oxidase, forming what? Gluconic acid and H2O2. H2O2 means hydrogen peroxide. Okay, now hydrogen peroxide is again oxidized by the peroxidase. See, with the name only, the enzymes are also similar. Hydrogen peroxide is oxidized by the peroxidase and forming what? H2O, that means water, plus O means nascent oxygen. Okay, nascent oxygen is highly reactive, which is reacting with the 4-amino antipyrin in presence of the phenol in the phosphate buffer. After this reaction, what is happening? Pink color chromogen is forming. Okay, and we are estimating by this color change. By this color change, you can understand whether there is a presence of glucose or not. If there is a presence of glucose, definitely this color change will be there. If there is no color change, means glucose is absent. Okay, that's all. Now this is your enzymatic reactions. Okay, so I'll come into that. Okay, this is enzyme reagent. These are the four things, and now you know how they are helping. First, GOD is glucose oxidase is oxidizing the glucose and forming the gluconic acid and H2O2, that is the hydrogen peroxide. Hydrogen peroxide is oxidized by the peroxidase and forming the H2O and the nascent oxygen. Now the nascent oxygen is reacting with the 4-amino antipyrin and the phenol and then forming the pink color complex. That's all. Done. Glucose standard sample is given. You know the concentration, and test sample you don't know the concentration. Now what to do in the test? First of all, we will take 3 test tubes. See here, these are the 3 test tubes. Okay, one is marked as B, another is S, another is T. B means it's a blank. Okay, standard means, S means standard and T means test. Okay, so from here you can understand that blank will be some control, standard is we already know, and test is the sample that we will be using to estimate the concentration. Okay, in blank what we will take? We will take around 1 milliliter of enzyme reagent, also known as the, let's say, GOD-POD reagent. Okay, enzyme reagent or the GOD-POD reagent we are taking 1 ml of it. Over here also 1 ml, over here also 1 ml. Okay, we took in 3 test tubes 1 ml of enzyme reagent. Done. In the blank we will add 10 microliter of distilled water. That is why it is known as blank. Okay. Over here we will add 10 microliter of the standard solution. Okay, that is why it is known as a standard. And test, we will add 10 microliter of unknown sample. Unknown test sample of the test serum. Right? Now you will mix all of them. Now, can anybody tell me what is the microliter value in ml? How much microliter is 1 ml? What?
-3.
I said how much microliter is 1 ml. I am asking the microliter value. 10 to the power 3. Yes, 10 to the power 3. So that means 1 ml equal to 1000 microliter. Okay, not -3. It will be 10 to the power 3 because I was asking the microliters value, not microliter to ml value. Okay, so that is what is written. Now when you will see the pipettes, you always have to look at the pipettes. How much is the valuation in the pipette? Okay, if it is written 1000 microliter means that is 1 ml. Okay, and that much amount you have to take in the 3 test tubes. Then another pipettes are there which is for smaller amounts of the pipetting. So over there you will fix like 10 microliters and you will take, first you will take the blank, that is the distilled water, then 10 microliters of the standard and then 10 microliters of the unknown sample. You mix it up, then you have to wait for 10 minutes. Okay, 10 minutes at 37 degrees centigrade, that is around room temperature you can say. Okay, and you have to incubate. You have to incubate. Incubate means in some labs you can say that a lab inside room temperature is low, okay, to preserve the things. Even these reagents are preserved at a 2 to 8 degrees centigrade. So immediately if you take out the samples from the freezer and you do the test, this color change will not take place immediately. You have to first bring the samples in the room temperature. So that is why we have to incubate the samples. Okay, we incubate the samples for 10 minutes at 37 degrees centigrade. Okay, after that you will see the color change. Okay, once you see the color change, you will see over here there is no color change because you did not add anything that contains glucose. There was only GOD-POD reagent and distilled water. Over here you will see color change and over here also you will see color change. Okay, because this test sample ऐसा कभी नहीं होगा कि किसी पेशेंट का टेस्ट ब्लड के 0 ग्लूकोज है. It can be less, it can be more, but it will be positive. Some color change will be there. Now with your eyes you can estimate one thing, that is, if this color is much more deeper than this, that means this has more concentration of glucose. That is why the color is also more and the concentration is also more, and if you check the absorbance, that will also become more. Okay, if this color is lighter than this, then most likely this has less concentration than 100. But still you don't know whether it is hypo or hyperglycemic condition. Okay, fine. Let us see what it's written over there. Take 3 test tubes. Take 3 test tubes. Mark them as B, S and T. Take 1 ml of enzyme reagent in all of them. Done. Then after that you have to add 10 microliters of distilled water, 10 microliters of standard and 10 microliters of sample, unknown sample in the BST. And then you have to mix it well and incubate it at 37 degrees centigrade for 10 minutes. Okay, and take the reading at 520 nanometers. Now 520 nanometers is the lambda max. You know the concept of lambda max? Beer-Lambert's law? The wavelength at which the absorbance is the maximum, right? So that is the lambda max. Lambda max is given at 520 nanometer. Now there is one catch, that when you are using the kits, I said like the enzyme reagent and the glucose standard will come as a kit. There are different companies of kits. Different kits are there, and in different kits these wavelengths will be mentioned as different. Okay, the one that we are using is the Coral kit. Okay, so in the Coral kit it is mentioned as 505 nanometer. Okay, but in the colorimeter we don't have 505, so we are preferring as 520, the closest one. Okay, that is what we are using. And the filter color, which is important for you to know, is the what should be the filter color. The filter color should be green color. Okay, with each and every wavelength the filters colors are also different, right? Sir must have told you in your classes. At each and every wavelength there are different color coded filters are there in the instrument. Each wavelength means particular filter. Through that particular filter only the light will pass. right? So at 520 nanometer the filter is green color filter, and through which only the color will pass, and in that filter only you will get the maximum absorbance, and that is what you are taking. Okay, so this is your method. OD is the absorbance. Did you do the class of third class, that B.L. Lambert's law? Over there what we did was, first we measured the lambda max. Okay, how we measure the lambda max? We are putting the distilled water in the instrument, then we are making it 0, right? After we set the 0, then we are taking the colored sample, right? That KMnO4 sample or the potassium dichromate, whichever sample you are using. We used KMnO4, potassium permanganate sample. So over there, that color coded sample you are putting into the machine, and then you are estimating the lambda at multiple wavelengths like 400 nanometer, 440 nanometer, 450, 500, 520, 540. Everywhere you are measuring the lambdas. Then after that, what you are measuring, that's in the screen, whatever you are getting, that is the absorbance that you are getting. That is the OD. Okay, whichever OD is the maximum in whichever wavelength, that is your lambda max you are getting. Okay, and the values that you are getting, that is the absorbance. Absorbance means the concentration, the cubate that you are using inside the instrument, the fluid will contain some concentration of the KMnO4, and the more the concentration, the more will be the absorbance of the light. Like let's say this is a cubate, okay, and this is the light source. From here, light is passing through this cubate. Okay, there is a particular filter that you have set. The filter over here is, I am saying 520 nanometer. Let's say this is a 520 nanometer filter. Okay, the lights are passing through here, going into the cubates, and there are small particles of the KMnO4. Okay, and these particles will do what? They will absorb the light, and less amount of light will pass from here. Some bit will get absorbed over here by the constituents, by the particles. The more the particles, the more will be the light which will get absorbed over here. So depending upon that, if this concentration increases, then the absorbance will also increase. Okay, so that is the exact concept, that A is proportional to the C. Okay, that's all. Now what is happening over here? You have to measure the absorbance, that is written as OD. How much amount is getting absorbed? The more is the absorbance, the more is the concentration of the glucose in the solution. Right? That's all. Principle. You can say this is a reaction that is happening in the test tube. Okay. First of all, glucose is there in the sample. Okay. You are mixing the GOD-POD enzyme reagent with it. First, the GOD of the enzyme reacting with the glucose, oxidizing the glucose and forming the gluconic acid and potassium. This is hydrogen peroxide. Okay. Now this hydrogen peroxide is getting oxidized again by the peroxidase, forming H2O and nascent oxygen. This nascent oxygen is reacting with the remaining two compound, that is the 4-amino antipyrine and the phenol, and forming the pink color constituent. And if there is no glucose, this pink color will not come, because this whole step will not take place. That is why when we are adding the blank, in the blank we are taking the distilled water, there is no color change. But in the standard there is color change, because the glucose is there and it is a fixed concentration, that is 100 milligram per deciliter. And in case of test, we don't know, but there should be some, either less or more, we don't know, but should be some. So because of that, pink color will be there, but it could be lighter shade of pink, it could be dark shade of pink. If it is a dark shade of pink, you can understand the glucose concentration is more. If it is a lighter shade of pink, you can understand the glucose concentration is less, probably less. That is why it's a lighter shade. That's all. Okay. Now the same procedure is written as a chart so that you don't confuse things. First of all, you take 3 test tubes, mark them as B, S and T. Either you can mark in the test tube or you can mark in the rack where you are taking the test tube. So 3 test tubes you are marking as B, S and T. Each of the test tube is filled up by the enzyme reagent. What is the enzyme reagent? The GOD-POD reagent. Take 1 ml in each of them. Okay. Then after that you take distilled water. How much? 10 microliters in the blank. Standard of 100 milligram per deciliter in the standard, that is also the same 10 microliters, and in the test tube, test sample, that is also 10 microliters. Done. In some books it is written as 0.01 ml. Okay, so don't get confused, that is the same thing. 10 microliters and 0.01 ml is same thing. Okay. Somebody, if have any doubts, this is 10 microliters divided by 1000 microliters. That is the ml conversion, right? So that comes 0.01 ml. Okay, in some books it is written as 0.01 ml. So either you write 10 microliter or either you write 0.01 ml, it's the same thing. After you do this process, you will incubate this mixture. After that you will take out from the incubator and you will see some color changer there or not. Okay. Now you have 3 samples B, S and T in the test tubes, right? Now from the colorimeter you know that it has a specific cuvette where the length is also fixed for each of the cuvettes, right? Now you will transfer these samples into the cuvettes. Okay, this B1 will transfer in 1 cuvette, S1 will get transferred in 1 cuvette and T1 will also get transferred in 3 cuvettes. Now you will put the cuvettes in the colorimeter. Okay, the same way you have put your KMnO4 sample, the same way you will put these samples. Okay, and see the absorbance. Before seeing the absorbance, you make sure do 2 things. One is over here there is that scroller with which you can fix the filter. Which filter you are using? You will fix the filter at 520 nanometer. Filter is fixed. Then there is this absorbance and you have to make it 0 with the distilled water. Alright, you have done your distilled water. After that you have done the normal zeroing. After you do the 0, then you have to put B, S and T. Okay, you will get some values in the absorbance that is written as ODB, ODS and ODT. Okay, 3 values you will get. Let's say for now I am just giving you an example, 0.11, 0.30 and 0.50. Let's say. Okay, I am taking the OD test is coming more pink. Okay, that is why the absorbance is also more. Okay, once you get this, you have to put it in a formula. That is this formula. Okay, the formula is what? ODT minus ODB divided by ODS minus ODB into concentration of the standard. What is the concentration of the standard? 100. So over here it will be 100. Now if you calculate, let's see from here what we are getting. 50, 0.50 minus 0.11 and divided by 0.30 minus 0.11 into 100. What comes? 0.39 divided by 0.19 into 100. After this, whatever you will get around something like around 205 or something. That is your answer. That is your glucose concentration. Got it? Test is simple. Calculation is one thing that you have to do on your own. Okay. If somebody is good in maths, too good. Not so good, you have to do it, can't help it. Some people might face some difficulties over here, but this is very basic maths. Okay, just minus it and multiply it, that's all. There is one more way of doing it. Okay, so I told you about the kits, right? So from kit to kit it varies. Okay, in some kit what they do is in the instrument we are doing the 0 with the distilled water, right? What we will do is we will make another 0 after putting the ODB sample. Okay, that ODB sample we are taking, we will put the ODB sample and then make the value as 0. That means this minus step you won't have to do anymore. Because if you are making the ODB as a 0, then the standard and the test values are already getting low. Okay, and because of that, if you are making the ODB as 0, you can that time measure as ODT by ODS into 100. Whichever process you want to do, you can do.
So how to set ODB at 0?
You put the ODB cuvette in the calorimeter and then make the whatever value you are getting. I said like around 0.11, right? Right now I'll make it 0. Instead of 0.11, I'll make it 0. So eventually when I'll get the value of ODS, that time that value will also be 0.11 less. When I'll get this value, that time also the value will be 0.11 less, because I have already made the ODB at 0.
So it is better to put ODB
at 0. So there are different types of kits. Whichever you like, you can perform. Either if you are feeling like no, I'll make the distilled water at 0 and then I'll perform the test, you can get the value, solve the problem and you will get the result. If you are thinking like no, I'll make the ODB as my result, put the ODB, make it 0, you will ultimately get the similar result only. That's all. You take 1 ml of it. Okay. Pressure is created. Put it in. Take the sample. See, over there some air came, so I have to deduct it. I cannot take this. There should not be any air. If you see already in your sample there is air, you cannot take it.
Here is there 41 91 to 100.
Fine. Okay, 3 samples taken from the GOD-POD reagent. Okay, now we have to take first the distilled water. Took the distilled water, take the test tube. First see from here how much is the amount. Okay, see this much amount you are doing with, that's all. Take out the test tube. Done. Mix it, keep it here. Now after doing this distilled water, I am supposed to change the tip. Okay, but I know that this is distilled water, so there is no contamination over here. There will be no chance of contamination. So I can use the same tip and I can take the standard. Done. Now see another process where I am putting the tip inside, releasing the sample. See, there is nothing. So there is a better way of mixture. So there is nothing, no chance of this. Probably your hand is shaking. So somewhere this amount can get into the wall and it did not get mixed only. So don't do this kind of things. If it is not mixing, then the test will not come. Okay. Now this is done. Now after my standard, I have to change this tip. Because right now I will be taking samples from the unknown sample. If it is containing less, it gets mixed with the standard, it can give you a false result. Release it, take a fresh tip, and I will take from the sample.
How did you release it?
Over here. If you press it, it will get released. Okay? Done?
This is the test.
This is the test sample. Done? Okay. Now again after the test you have to discard. Why? The next person will come and again do the blank. So that's why blank should not get mixed with the test. Okay. Now my mixtures are done. I'll put it for the incubation. Okay.